To introduce Nicole Steinmetz. She's coming from San Diego, so a little bit more sun than we have here. Although we had also quite a lot in the summer now. And she will tell us something about plant virus nanoparticles for in situ vaccination treatment of metastatic cancer. Let's see how plants get along with humans or animals or cells from other sources.
Yeah, well, thank you for the introduction, and thank you also to the organizers for the kind invitation. It's great to be here, and yeah, I'm learning a lot about other systems than plant viruses. So yeah, we are working with plant viruses as a nano platform technology. So our research starts out in the greenhouse, or indoor greenhouse facilities where we grow cowpeas and Australian tobacco. And we use these plants merely as a means to produce nanoparticles, basically plant virus-based nanoparticles. So when I look at a virus, and this shows here the structure of the cowpea mosaic virus, all I see when I look at the slide is a platform technology, a tool that we can engineer to impart new functionalities targeting human health as well as plant health. So why have we chosen a plant virus-based nanotechnology? Well, personally, I think there's probably 101 good reasons. To go over a few, well, if you think of it, a virus naturally is a nanoscale platform. We can look at them as prefabricated nanoparticles. They're naturally expert at targeting cells, getting into cells, and delivering cargoes, their genomes. We've turned towards a plant virus-based platform versus a mammalian system, because there's an added layer of safety. Plant viruses are non-infectious towards humans. We have a high-precision nanomaterial. Essentially, each particle is a clone of each other. So from a quality control assurance point of view, you have a very high degree of a precision nanomaterial, very monodisperse and uniform. These materials are scalable and programmable, so we produce them using plants. On the left is shown a black-eyed pea plant that's infected by mechanical inoculation. So you can tell these are highly sophisticated techniques where you take purified plant virus particles and then you gently rub it onto the leaves. The leaves are dusted with carborundum, a fine silica sand, to allow to create some lesions in the leaf, allow the virus to enter and then spread through the plant. For translational purposes, we also use agroinfiltration techniques to make VLPs. So these are virus-like particles that lack the plant virus genome. So they are non-infectious also towards plants. And we can produce these materials in very high yields. So if I set up 30 plants, that would be the size of this podium here. I would generate about 100 grams of infected leaf material, and we can extract up to 500 milligrams of pure viral particles from that amount of leaves. So in my lab, we use plant viruses as a tool for various applications, ranging from molecular imaging and risk stratification to drug delivery, targeting both human health as well as plant health. So we are repurposing plant viruses to deliver pesticides into soil. And what I want to talk about today is some of our research in the area of immunotherapy. I don't think I have to give too much of an introduction, but just to put this into context, we're working on in situ vaccines. And the principle of an in situ vaccine is that you take an immune stimulatory agent, in our case a plant virus, and we inject it directly into an identified tumor to essentially restart the cancer immunity cycle. The cancer immunity cycle is depicted here. What happens is once a tumor cell undergoes apoptosis, this tumor cell would shed antigens. These antigens are recognized by dendritic cells. Dendritic cells talk to T cells, and the T cells are then primed to recognize and eradicate this tumor. However, the cycle, in theory, is a great cycle and the body's defense against developing tumors. However, over time, the tumor will essentially shut down this cancer immunity cycle by presenting an immune suppressive tumor microenvironment by a number of mechanisms. So what we've thought to do is to take a VLP, a plant-derived VLP, to restart the cancer immunity cycle. And I want to point out what we're using here is the cowpea mosaic virus, or VLPs thereof. They are not modified at this point, so we look at them as our generation 1 particle. We haven't yet added a drug or another immune stimulatory agent. We're just making use of the multivalent protein structure of these particles. So we tested this concept of in situ vaccination in a number of animal models. Shown here on the top is a breast cancer model, so that's a metastatic lung model where we take 41 cells, we inject them intravenously, they will then home to the lungs. And you see on the right is a PBS-treated animal where you see heavy disease burden within the lungs. The eCPMV treatment group, so eCPMV is essentially just the VLP of the cowpea mosaic virus. We injected those intratracheally. So the idea is that we're treating the lungs, or which present the tumor, and we see that we have 50% survival. We can do this with melanoma grown in the skin, or sub-Q tumors, shown here is a colon cancer. And at the bottom, an ovarian tumor model where we have IP disseminated disease. We inject the particles, also IP, and we see we have about 50% survival. The immunotherapy is both local as well as systemic. So if I have an animal that has a primary tumor in the skin, and I'm only treating the skin, I will see the disease regression both in the skin as well as the metastatic disease that might present within the lungs also regressing. More recently, this is some unpublished data. We also looked at maybe more challenging to treat tumors. So this is a glioma model where you see on the left, this is a mouse with a glioma in the brain. And then the CPMV treatment group is disease-free. And again, we have about 50% responders and 50% survival. We repeated those studies in a number of knockout models, demonstrating that this is indeed, the anti-tumor efficacy is indeed immune-mediated. We see a downregulation of MDSCs. We see an infiltration of innate immune cells, and particularly neutrophils. These tumor-infiltrating neutrophils are antigen-presenting and T-cell priming. So what we're essentially doing is we are reprogramming the tumor microenvironment to get an infiltration and activation of the innate immune system. We then get a crosstalk to the adaptive immune system, leading to a systemic anti-tumor immune response. So not only do we see treatment of disease as well as prevention of outgrowth of metastatic disease, we also see that we have an immune memory effect. So here's a mouse model where we grow a dermal melanoma derived from B16F10 cells. These tumors are treated by intratumoral injection directly into the tumor. Again, we have about 50% survival in this animal model. If we then re-challenge the survivors with the same tumor, the tumors essentially don't grow. And so this attributes to this immune memory effect. This is also reproducible in other tumor models, so this is more recent data, not published yet, looking in the ovarian cancer model, where again, if we take survivors and we re-challenge them with the same tumor, the tumors essentially don't take, and this can be attributed to production of CD4, CD8 positive cells, as well as effector memory cells. So the way we understand this in situ vaccination with the VLP so far, I mean, there's still a lot of open gaps and questions we need to address, but we take particles, we inject them directly into the tumor. This will reprogram the tumor microenvironment locally, lead to the infiltration and activation of neutrophils that will eventually recruit T cells, and that these T cells provide systemic anti-tumor immunity. So we've done this with CPMV, cowpea mosaic virus. Now the question is, can you take any plant virus, inject it in a tumor, and you get this almost magical anti-tumor effect? And the answer is no. So we've tested the CPMV in comparison to other adjuvants, so looking at poly-IC, a STING agonist, and green DMXAA, as well as LPS. And we see in each case that the CPMV particle outperforms other adjuvants. When we compare it to other VLPs, here's just a few particles that we recently tested, CCMV, SCMV, PHMV, all particles that are similar in size and similar in structure, but none of these particles really induces this anti-tumor effect. So obviously the question is, what's so special about the CPMV, and why does it outperform other adjuvants and alternate VLPs? Well, I wish I could give you the answer at this point. We have a large program now to probe this question and to delineate what is so different about CPMV that primes this very effective response versus other VLPs. I don't know how much time I have left. 2 minutes, okay. So moving forward, so one direction is obviously to go down and study the mechanism, dive down in the mechanism of this response. But the other arm that we're pursuing is the translational development. So what we found is we do need to inject these particles multiple times into the tumor. Generally, we treat over a time course of 2 weeks using 2 to 4 injections to get an efficient response. Now that's fine for some tumors, but some tumors, other tumors will be difficult to treat that way. So for example, a glioma, I discussed this with several brain cancer surgeons, and they all tell me you can't take a patient and inject them 4 times in the brain. Fair enough. So what we're doing now is we're assembling these materials essentially into assemblies and into hydrogels that we then envision we could implant post-surgery into the tumor site, and then because there will still be some residual disease, and then reprogram the tumor microenvironment of the residual disease to get this anti-tumor effect. So we're looking at crystalline assemblies, films, as well as polymer processing to make implants and microneedles. So here's one data set where we actually used an ovarian cancer model to kind of prove this concept. So if we take the particles and we only inject them once, we essentially don't see any efficacy. So that would be in panel B, if you look at the green line, so that's the CPMV particle at a high dose injected once, so there's no efficacy in reduction of tumor burden. But if we take our assembly, that would be the purple line in panel A, CPMV G4, if we inject this only once, we do see we have the same efficacy as if we treat weekly with these particles. And we can explain that by an enhanced residence time within the tumor microenvironment. So making these hydrogels, we might be able to also treat tumors that are challenging to inject multiple times. Another direction that we've taken is to work together with a vet at Dartmouth Medical School. So this is a collaboration with Steve Firing and Jack Hoops, who are both at Dartmouth Geisel School of Medicine. And we started treating companion dogs. So this is not a dog model. It's essentially somebody's pet developing spontaneous tumors. And so here's one example where we treated a beagle who came in with a 5 centimeter large melanoma that penetrated the nasal cavity. So these are oral melanomas. And in this specific animal, the clinical decision was to treat with a combination of radiation and the VLP and Situ Vaccination. So you're seeing images here before and after treatment. So this was a 2-week treatment schedule using fractionated radiation and the VLP injected directly into this tumor. And even after 2 weeks, we see a dramatic reduction of tumor burden. And if we look again after 6 months, there's essentially no tumor and no signs of metastatic disease. To date, we have treated 4 canine patients. The patient shown here, Paige is her name. This patient is still in complete remission 22 months post-treatment. And this is quite an amazing result. When I talk to the vets, they say when they come in with this high disease burden, the survival time generally is 6 to 8 months. So again, so far we've only treated 4 patients, but all 4 patients responded well to the treatment and are in complete remission at least 10 months plus post-treatment. So we can't make too many claims. It's not enough to do statistics, but so far I think the data are very promising. Yeah, so with this, I'd like to come to an end and thank my group. We just recently moved to the University of California in San Diego. So I have a group of scientists, postdocs, graduate students who do all the work. I just do all the talking. And our funding sources from NIH, NSF, and the American Cancer Society. Thank you very much.
Thank you, Nicole. Great talk. Questions? Rudolf?
I hope that I did not miss it, but the thing is, you just always took for natural virus particles. what do they have in them, DNA or RNA?
So in this case, we used a VLP, a virus-like particle, and they're devoid of RNA and devoid of DNA. So it's just a protein capsid.
Okay, now the thing would be, one end would be that I was thinking that if it would be even stronger as an activator, is it contains some RNA? Because
yes.
I think when this all goes into the fear of the body of a virus attack, and the other thing is, how easy is it to make some chemistry? Or for example, if you fill it again, can you put some antigen into the virus?
Yeah, so we looked at studies recently where we also compared it systematically with the native particle containing a single-stranded RNA genome, and the efficacy indeed is more profound, but not by much. Like we can put an error bar and statistics on it and show it's statistically significant, but from a clinical perspective, the outcome or survival isn't that much enhanced. The chemistry is well established, so we can either use chemistry covalent conjugation to conjugate antigens on the inside, the outside of the particle, use genetic fusions to incorporate specific antigens as well, so we can put in peptides as well as chemical drugs. So we looked at combination with chemotherapies, specifically chemotherapies that induce immunogenic cell death. But we found that if we combine it, so if we attach it to the particle, it's actually not necessarily more active, but if we combine it and inject these things separately, then we get a synergistic effect.
Yeah, really amazing talk. Do you have any clue on the underlying molecular biology reason why these virus-like particles are so tremendously active?
Yeah, we're starting to get some clues. So the receptors that are being recognized are TLR receptors, TLR2 and TLR4. The signaling seems to be MyET88 dependent, which kind of makes sense if it's TLR and a protein-based platform. What's still puzzling to me is why is the cowpea mosaic virus so much more potent than the other platforms? And we are now starting to also look at bacteriophages as well as VLPs of AAV to just get some comparison, but also hopefully diving into the mechanism. We have one hypothesis is that it may have to do with vimentin binding. So vimentin is a cytoskeleton protein, but also a secreted protein that's secreted by neutrophils, has been found in some tumors in the extracellular matrix. And even though a cowpea mosaic virus is a plant virus, it has been shown to bind to mammalian vimentin. And there are some structural similarities between CPMV and polio, as well as tyler-myelitis encephalus virus that explain this sort of molecular interaction. And so what we're doing now is we take particles that don't have efficacy and we include features of the cowpea mosaic virus that we think render them vimentin specific to see whether that has anything to do with it. And we have some data on it, but they're not that conclusive at this point. It's like one day I go home thinking it is vimentin, and one day I go home and think it's not vimentin. So it's too early to really draw any conclusions. Thank you.
So I have a question on blood-brain penetration. Are these viral particles penetrate the blood-brain barrier or not?
There are some, so in this case we injected them directly into the tumor. There's a couple of papers where data indicate that they do penetrate the blood-brain barrier in diseased models. So in a tumor mouse model or mouse models of brain inflammation. However, I also think in those mouse models, the blood-brain barrier is probably already compromised, therefore allowing the viruses to cross. There's some claims in the literature that plant viruses, especially CPMV, can cross the blood-brain barrier also because of this vimentin interaction. But I'm not sure that that holds true if you would take a mouse model with a non-compromised blood-brain barrier.
Yeah, and the effects that you see, is that some similarity with Zika virus and glioma treatment, where you also see effects? And
yeah, we haven't. We just moved to UCSD, so did The PI who did the studies was Zika virus in the glioma model, Jeremy Rich. So I haven't met him yet, but I'd like to meet with him and see if we can do a study side by side. I think the Zika virus still has a different, well, maybe the immune activation is a similar mechanism, but I think that was systemically administered, and then they make use of the fact that these viruses specifically target the cancer stem cells within glioma.
Okay, thank you.
Yeah.
This question here in the middle.
I just had two little questions. One was regarding, as you probably mentioned at the beginning, also the purity of this. Is there any endotoxin contamination once you extract this?
Yeah, so we've looked into this quite a bit, and there is generally no endotoxins coming from the plants, from the methods. But what we found is you have endotoxins in your lab, and it's really hard to get rid of them. But we have, as part of our purification protocol, we always check for endotoxins and go through endotoxin removal steps. I find all these protocols are really low yielding, so we extract a lot of particles from the plants, but then before we really purify them to go into the animal study, and not so much from the plant contamination, just like from working in a non-clean room lab. I find like we lose a lot of material in the process, but they're all endotoxin free before we inject them into animals.
And just the second one I just had about, since your source is proteinic, made from protein, I was wondering if you also obtain an immunity against the virus. So in terms of if you would apply it later on to a different tumor, for instance, then this method would not be applicable anymore.
We looked into this, and essentially pre-immunized mice just by sub-Q administration was an adjuvant to generate high antibody titers against these particles in mice. Then we injected the tumors and then did the treatment. And if anything, the response becomes more potent. So what we think is happening, if you have antibodies already present and you inject the particles in the tumor, the particle gets coated, and via the Fc part of the antibodies, the immune cell interaction is even more efficient.
So it's the other way around, though?
Yeah.
Okay.
Nicole, I have a more general question. So if there would be a very rich investor that brings you a ton of money, which particle would you go for, in which cancer setting, in what combination treatment? What would be your lead way to go forward translationally? Yeah,
so I think definitely the VLPs of the cowpea mosaic virus, because we've shown potent efficacy across tumor models, including in the dogs. For the tumor model, so we would set up a trial with cutaneous injectable tumors. So that would include melanoma. There are some recurrent, difficult to treat cutaneous breast tumors, as well as anything that's injectable cutaneous.
Would those be the ones that do not respond to regular CTLA-4 inhibition or PD-1 inhibition? And is there good reason to believe that your particles will be able to add from cold to hot tumors? Because I think that's what everybody's looking for, right? Yeah.
So we have some evidence that PD-1 or PD-1 is actually upregulated. Yeah, PD-1 is the one on the tumor cells, right? Yeah. PD-L1. Yeah, PD-L1 is after we treat tumors with our particles, we see an upregulation of PD-1, and since we also see a recruitment of T cells into the tumor tissue, that the combination with the checkpoint inhibitors may be beneficial. We've done some initial studies using CD47, which targets the sub-alpha axis, and we see that we do see synergy combining those two, but we haven't looked at like CTLA-4 or PD-1.
Sounds good. Last chance for a question. If not, let's thank Professor Steinmetz again.